Hi Year 10s, this is the second video in our cell division topic, so week 2 of our Year 10 trimester. We're looking at cell division, we're specifically looking at mitosis in this video. It is important, the first slide that we go through you don't have to draw the picture, but for the future slides for mitosis you will need to draw the pictures because you do need to be familiar with each step in mitosis, being able to talk about it and draw about it. So, first off, I suppose the stages of mitosis, and I suppose what even is mitosis. So mitosis is the breaking down of the nucleus, the separation of our replicated chromosomes into single-stranded chromatids. The steps of mitosis are prophase, metaphase, anaphase, and telophase, and you can remember that with the acronym PMAT. Okay, so PMAT, prophase, metaphase, anaphase, telophase. Moving on, we're going to start by looking at prophase. So in prophase, chromosomes condense, which means they get small, they get a lot more coiled up, and because they're getting coiled up, think of it like a sheet of paper that's being held up like that. But if we crumple it up as a fist, that becomes like thicker and more visible, even though it's smaller. That's what's happening with the chromosomes. So they're condensing and they're becoming visible with microscopes. We can now see them. Centrioles, which are cylindrical structures of protein, which we can see here in the brown, they are moving to opposite ends of the cell, called poles, and they produce something called spindle fibers. So we've got the centriole, which is the little circle, and the spindle fibers are the lines that are coming out. It's also important here, I haven't actually written in the text, but the nuclear membrane, which is around the nucleus and containing the chromosomes, that's breaking down. So you can see that there. I've drawn a dotted line to show that that's breaking down. So basically, for prophase, the first stage, nuclear membrane is breaking down, chromosomes are becoming visible, our centrioles are moving to opposite ends of the poles, and we've got the spindle fibers being formed. We then move into the metaphase. This is the second phase, and in this one, the spindle fibers are contracting. So you can see that the spindle fibers have attached to each, both of the spindle fibers from both ends have attached to a chromosome. And then they're both pulling with exactly the same force. So as they're pulling with exactly the same force, what happens is the chromosome gets pulled to the dead center of the cell because they've got equal force on both sides. They're getting drawn to the both poles with equal force, which results in them going to the center of the cell. That happens to all of our chromosomes. So you can see in this stage that all the chromosomes are lined up down the center of the cell, being pulled by the spindle fibers from the centrioles. Easy way to remember this stage is metaphase, M, middle, all the chromosomes being lined up down the middle. We then move to anaphase. And in this stage, the spindle fibers are still contracting, they're still pulling with force. But unfortunately, at this point, the chromosomes can't hold on any longer. That forces the chromosome to split, and the two replicated chromosomes separate so that we have one single strand of chromosome on each side of the spindle fibers, being pulled to opposite ends of the cell. We can also see that the cell itself has started to elongate, getting ready to separate into two different cells, which is what's going to happen in telophase. At this point, now that we have our chromosomes that are single-stranded, a nuclear membrane forms around them. So we have our nucleus with our chromosomes in there again. The cell itself, the original parent cell, has elongated to the point that the cell has now split into two, and we have two identical cells, both of which are identical to the parent cell at the beginning before DNA replication took place. So if we go back to that first stage in prophase, the chromosomes do look different between prophase and telophase in my drawings. The reason for that, in prophase, DNA replication has already taken place, which is why we have them looking like this X shape, because DNA replication has taken place and the DNA has been duplicated.